So our next speaker is going to be Juan Marcos.
Thank you. Good morning, everyone. Very excited to be here. Thank you to all the organizers. I prepared a 10-minute script. I don't think I'm going to follow it, but I'm afraid of the bell. I think I have to start with a big apology because I'm not going to talk. I'm going to do an extraordinary effort not to talk about long-term pollination or anything about that. So probably you want to know about that, come to my laboratory. I want to tell you about a project, a particular project among the different projects in my laboratory, when we try to figure it out, the organization of the memory engram. And when it comes to the understanding the memory engram, we have the very easy definition and a difficult one. The easy definition, it is all the changes that happen in your brain that are made by memory when you acquire information. The difficult one is, what are those? So Doc Sachter is doing a great job to figure that out. And where do they happen? And that is my question. We want to know what is the architecture of the memory-associated changes in the brain across different levels of information, if you will. And I want to present to you a quote that I like very much. It was developed by Isaac Bingham. I'm going to tell you about that later, one of the students in the laboratory. It seems as though such an architecture, the one of the memory engram, across different levels, must be dictated by an underlying structure. So one basal instruction feed the other one and the other one and to the other one and to higher levels. So I promise to you to tell you a little bit about an approach that's going to be very superficial. So stop me if I'm going too complex in my explanation. We focus on we focus on the hippocampus and its structures in the middle temporal lobe, highly associated to memory and the maintenance of memory, not all memories, but spatial and declarative memories. The most important point that I wanted to give you here is that from studying the hippocampus, great scientists have formulated one of the most dominant hypotheses in the field of neuroscience memory, which is the synaptic plasticity hypothesis of memory. It reads that neural activity that underlies experience changes the efficacy of appropriate synapses, that LTP that Todd was talking about, to create and store memory for long term. So we look into the hippocampus and we wanted to figure it out, what are the changes in this synaptic transmission, synaptic efficacy across the hippocampal circuit. And the hippocampal circuitry looks very daunting in here. That's what I really needed where is the cursor?
I'd have to get the cursor here.
Are you guys got that from my time? Yes. All right, thank you so much. Because it's extremely important that I have the cursor in here, because this circuitry you can see in here, the anatomical cartoon, the gyrus areas, CA3, CA2, CA1 areas are represented in here, the gyrus, CA3, CA1, and all the arrows represent synaptic relays. I want to reduce this very complicated circuitry into two separate information streams. One of them, I call it the inner loop, which are the synaptic relays from the data gyrus into the hippocampal proper, CA3 to CA2, CA1, and then goes back to the cortex. This is the inner loop. And then I want to make a separate distinction to what I call the outer loops. The outer loops are the one synaptic relays from extra hippocampal regions, mostly the entorhinal cortex, that goes to every one of these hippocampal areas, but separately and independent. So you can see that we have a balance between two different information streams, the inner loop and the outer loop. So what we did was to look into some of the synaptic relays that belong to the inner loop and outer loop after memory. We trained animals in the Fenton task, the beautiful task that Andre presented. It's a cognitive task. And we did it in different regions, and the first one that we did was in the distal dendrites of the dendrogyrus synapses. So that's one of the relays. And what you see here is a plot that represents here synaptic transmission strength. Here is the amplitude of the strength, and here is the stimulation, so stimulus response. And red will always show you the memory trained animals, and black the control untrained animals, the ones that actually are there foraging around, but they're not being memory trained. And you can see, interestingly, that if you look at the synaptic response in the distal dendrites, in the distal synapses in the dentate gyrus, you have a decrease of synaptic efficacy, a decrease in synaptic transmission. Unexpected, because you were expecting this long-term potentiation. Then, if you look into the proximal dendrites, the ones that are closer to the cell body in the same area, you see an increase in synaptic potentiation. So now we jump into the CA1 area, here, the exit relay, if you will, in the hippocampus, and you can see that we interrogated the proximal dendrites in the CA1 area. These synapses receive information from the inner loop. It receives information from CA2 and CA3. And you see the expected increase in synaptic transmission. And then we interrogated the outer loop, which is at the distal dendrite, and we found no change. So we're really excited about what's going on with memory and different stages of memory in here. But the main talk message in here is an speculation, an hypothesis in which memory-associated surface changes can fine-tune synaptic weights based on experience demand. This is happening after an animal has been trained. But we wanted to move forward, and thank you so very much, Andrew Fenton. Thank you so very much, Top Sector, and thank you, Andrew Sherepov, because thanks to them, we actually got our hands into this beautiful transgenic animal that Andrew introduced. You can see in here that it's an immediate early gene tagging system, in vivo immediate early gene tagging system. What does it mean? It means that when the animal is moving around and there's a strong neural activity happening in these neurons, you activate a promoter, in this case ARC, which is related with synaptic plasticity and memory, and you can produce expression of this reporter, EYFP. So you can see the neurons that are activated by the memory ensemble. Now, the great things about this is you can control the expression of EYFP by a Cree recombinance. That means that whatever I say, or actually whatever my students say, they can activate the expression and tag at different memory epochs. When the animal is learning something, when the animal has learned something, or a month after learning something. So we did that, and you can see here, cartoon represented the activation, recruitment, if you will, and we measured the fractions of activated neurons in the dentate gyrus area and the CA1 area and other areas, but at different epochs. We have the untrained animal in here. We have a home cage animal in here. We have an animal that is just learning the task, hopefully right now you guys in this moment, and animals that have learned the task, probably you at 6 p.m. today. So what we found is an increase in the recruitment of cells in the dentate gyrus at the level of the upper blade. You see, during learning, they have this big increase. This means the fraction of neurons activated. But then the increase is maintained, but it's not as robust when it comes to the maintenance of that. And then we have, that was 2 minutes or 1 minute?
30 more seconds.
What?
20 more seconds.
Okay.
A minute. Take a minute.
A minute, I'm going to take a minute. And then no change in the lower plane. In CA1, we also see an increase in neural activation and even a larger increase in the memory trained animals. And I really appreciate the 40 seconds that I have left because I really want to tell you what I really wanted to come in here, which is the work that is being done by my collaborators in here at SUNY Downstate recently. I have to mention Oleg Ebrakov, Ivan Hernandez, and graduate students, Isaac Bingham. What he did, he used spatial transcriptomics to look at the molecular footprint of changes associated with memory. In here you can see a UMAT expression, a gene expression map that nicely mapped to the anatomy of the hippocampus. And what Isaac found, and this is my last slide, is when he did RNA sequencing analysis and then map the gene expression in animals that have been trained and map it back into the anatomy of the hippocampus, he found that the main biological process being activated is the regulation of transsynaptic signaling. And if you ask the machine which gene is the best expressed in this process, it was HOMER3. And why is HOMER3 relevant? Because HOMER3 is a scaffolding protein, one that whose work is to ship materials, products from the from the dendrite to the synapse, similar to our friend Kybra in here. So you might be actually on the right track. So, thank you so very much to all of you, and thank you for
Okay, thank you, Juan Marcos.